Good morning, everyone. My name is Mike Brown. I'm a principal with Dowling and Yahnke, and I'd like to welcome you to the second in our webcast series on COVID-19, which will focus on COVID-19 testing. We've done a number of educational events over the past few years on health issues, and none is more relevant today than COVID-19. We're going to be recording today's session and accepting questions, so please feel free to submit your questions throughout the webcast, and we'll do our best to address as many as we can. At Dowling and Yahnke, our focus is to help clients make great decisions, and the more we know, the better decisions we can make. Our goal this morning is to learn more about COVID-19 testing so we can make more informed decisions about our health and well-being. We're blessed to have a great network in the biotech community in San Diego, and this morning we'll be hearing from Marco Biamonte and Melissa Chalmers of Drugs and Diagnostics for Tropical Diseases, or DDTD for short. Marco is DDTD's CEO, and Melissa is the Executive Director of Development for DDTD. So DDTD typically focuses on developing easy-to-use, rapid, inexpensive diagnostic tests to detect the causes of diseases such as river blindness, filariasis, and lowiasis in low-income areas. If you're not familiar with these diseases, I'd encourage you to check out DDT's website to learn more. In short, DDTD helps develop inexpensive tests that have a huge impact on the lives of those who receive them. So with that brief introduction, I'm going to turn it over to Melissa Chalmers, who's going to talk about the innovative work that DDTD is doing in the area of COVID-19 testing. Melissa?
Thanks, Mike. Can you hear me okay?
Yes.
Great. So thank you for the wonderful introduction. I'm just going to share a little bit more about our organization, and then I'll hand things off to Marco for the presentation. So Drugs and Diagnostics for Tropical Diseases, or DDTD, as Mike mentioned, we are a 501c3 nonprofit based in San Diego, California. Our mission is to develop treatments and diagnostics for vulnerable low-income populations, whether domestically or abroad. Our work is primarily focused on diseases that affect people in Africa, Asia, Pacific Islands, etc. And Mike had actually included some of those diseases. However, we are now also working on COVID-19, as well as have plans to tackle other diseases that affect predominantly homeless people in the US within the next year. The bulk of our work is to tackle infectious diseases that in the US we rarely hear of, such as river blindness or elephantitis, but which affects tens and tens of millions of people, usually in the most destitute countries. To combat these diseases, the World Health Organization has been implementing for decades mass drug administration programs, distributing hundreds of millions of preventative medications year after year around the globe to eliminate these so-called neglected tropical diseases. In order for these programs to be the most effective, you need to be able to map the disease before you start an intervention in order to understand the landscape of the reach of the disease during the intervention so that you can monitor progress and at the end of it as an intervention to ensure that the disease has indeed been eliminated and that there are no hotspots left. Anytime you need to map a disease, you need diagnostic tools to test for the disease, and that's where we come in. We specialize in high quality, low cost, rapid tests for diseases that primarily affect impoverished populations. Our funding primarily comes from organizations such as the Centers for Disease Control and Prevention, or the CDC, the National Institutes of Health, the Foundation for Innovative New Diagnostics in Geneva, Switzerland, a large group connected to the WHO, the Wellcome Trust, and philanthropists in the San Diego area. I'd now like to introduce our CEO, Dr. Marco Biamonte, who will be giving the presentation today. Marco holds a Ph.D. in organic chemistry from the University of Cambridge, worked as a medicinal chemist in the pharmaceutical industry for 14 years, and advanced 2 new drugs into clinical trials before starting DDTD in 2011. Marco is considered a subject matter expert on neglected tropical diseases, or NTDs, and is a member of the WHO Diagnostics Technical Advisory Group for NTDs, as well as a grant reviewer for the European and developing countries clinical trials partnership with the European Union for COVID-19 in Africa. And now I'll hand it over to Marco.
Thank you, everyone. Let me first pull up my slides. Let's start with that. And can you see the slides coming up?
Yes.
Okay. And I'm going to bring up the pointer. There we go. So, yeah, I would like to thank the organizers, of course, Mike Brown and Kathleen Arciniega, as well as their institution, Dowling and Yahnke, Wealth Advisor, for the opportunity to present, and Mike, thanks to you for your constant support over the years. I should add to all the attendees that I wish I could see your beautiful faces, but I cannot. That's a little distressing, but hopefully one day we'll be able to talk face to face. So my objective today is to give you a crash course so that you may understand the basics of testing for COVID-19. And I need to be upfront, I do not have all the answers, and no one does, not even the very best experts. But anyways, we'll start with a brief overview of the virus, and then we'll discuss a couple of use cases. For instance, how do the needs of a medical doctor differ from those of a government official? Then we'll look a little bit into the aspect of these tests and their accuracy. And finally, I'll say a few words about our approach and do my best to answer your questions. But let's start with some basic nomenclature. So SARS-CoV-2 is the name of the virus, and COVID-19 is the name of the disease that results from an infection with said virus. This is very much like HIV versus AIDS. HIV is the name of the virus, AIDS is the resulting medical condition. So by now, everybody should have seen somewhere on the news or wherever a picture of the structure of the SARS-CoV-2 virus. It's made of just 4 different proteins. That's not much, but there's multiple replicates of each. The red spikes here serve to attach to human cells and invade those human cells. And the gray envelope here encapsulates the virus genetic material, which is called RNA. And for all practical purposes today, just to keep things simple, we'll just say that viral RNA is very similar to the human DNA. Now, humans have a sophisticated machinery that reads the DNA and that synthesizes the protein accordingly. So you know that there's a certain code containing the DNA, then there's a number of biochemical transformations that must happen, and finally a protein is churned out of which the sequence reflects the code of the DNA. So humans have this machinery, but the viruses lack it, and they cannot replicate alone. So in order to proliferate, they are obliged to hijack the human machinery. They have to enter into a human cell and inject their RNA into the human cell. So on this cartoon, this box here represents a human cell. The virus arrives, gets in, releases its RNA, and then the human machinery takes over and generates viral proteins, which then self-assemble to give birth to a new generation of viruses. So from, I was forgetting to say that in the meantime, as these proteins becoming more abundant in the body, humans start fighting back with antibodies. So they will develop antibodies against the virus, and these antibodies take anywhere from 1 to 3 weeks to appear, and then they persist for months, even once the infection has cleared, with the goal to protect the person against future infections. For those of you who are number-oriented people, 30% of the people develop antibodies 1 week after the initial symptoms, and then the number goes up progressively. About 80% have them after 2 weeks, and over 90% of the people have antibodies after 3 weeks. And the antibodies look like this, with this characteristic Y shape. So from a diagnostic development perspective, this has the following implications. Diagnostics can be made to detect 3 different things. You can detect the viral RNA, You can detect the viral proteins, or you can detect the human antibodies that appear in response to these proteins. So if we're detecting viral RNA, it's called a molecular test. It's also called sometimes a nucleic acid test, NA, like in RNA. And this includes techniques like PCR, which is by far the most popular one. If you're detecting viral proteins, it's called an antigen test. And if you're detecting human antibodies, well, it's called an antibody test. Or a synonym for that is a serological test. So a serological test detects human antibodies. So let's try to put that in the context of a couple of different use cases. The very first one is the one of a medical doctor. So the medical doctor wants to know if the patient that he or she has in front of him or her requires a treatment. That's one possibility, but also another possibility is that the doctor needs to admit patient so-and-so into the hospital for whatever reason, a broken leg or whatever it might be, but they also need to know if the person has COVID-19 because they need to know if they can put him or her close to other patients. So what the medical doctor needs is really the ability to detect an active infection. And for that, they need to detect either the RNA, so they need a molecular test, or they need to be able to detect the protein, so they need an antigen test. What's not appropriate for a medical doctor is an antibody test. For two reasons. The first one is that the antibodies appear late in the infection, 1 to 3 weeks after the first symptoms. So by the time people arrive at the hospital, they may not have antibodies. And then the antibodies remain in circulation even once the person is healed. So you have no way of saying if the person who has antibodies was infected 3 months ago or was infected just last week. Now, I apologize, we are close to the Miramar Air Base and we have some F-18 jets flying over my head. I'll pause for a second. And let's try to resume. So the second use case is very different, and I think it's a very interesting one. It's the one of the government official. So the government official is in a very different position than the medical doctor. The government official does not care about individuals. It doesn't care about whether they're infected really at the time of testing. What he does care about is about populations, so groups of people, and he or she needs to be able to make public health recommendations. So for instance, questions that might float through the mind of some official are these. Have cases in San Diego schools been underreported? Or what about Tijuana? Tijuana is not reporting a ton of cases, but is this being underreported? And should one be more vigilant? Or alternatively, there can be different questions. For instance, should there be different rules for different industries? Should we relax different industries at different paces? Should you have different rules for the meat packing industry versus other sectors of the economy? So these are all very valid questions, but they require garnering data on a group of people. And for that, an antibody-based test is probably your best option because the antibodies give the history of a population. So let's take the example of a San Diego school. You may take a school of 100 students and go and test them today, but you might not be able to find a single case of active infection by molecular test, just because everybody's been self-quarantining in the last 2 weeks. But an antibody test may still reveal if X% of the student population has been infected at some point in time. And so that's useful data. It does require a bit of mental gymnastics, but it's useful data for, again, an epidemiologist or a government official. The third case is, of course, the one we're all concerned about. It's just you and I, right? So questions like, have I had symptoms in February? I'm curious, was it really COVID-19? Well, you need to have an antibody test. The problem is that they're not yet allowed for at-home testing. Or alternatively, I'm feeling fine, but could I be asymptomatic? Do I pose a risk to my family and colleagues? Well, here, a molecular test would be ideal, but again, they're expensive, they're a hassle, especially if you want to do that on a weekly basis. They need to be done by trained personnel. They can't be done at home. And so the bottom line is that currently there's really no convenient solutions. There's only cumbersome roundabouts. And I'll just introduce a fourth use case, is the perspective of the employer. The employer has many employees, some of which must meet with customers face-to-face. Maybe I own Disneyland. What do I do? How do I manage this responsibly? Well, a molecular test should be done on a frequent basis, maybe by spitting in a tube and sending it to a lab. And then on a more occasional basis, you do want to have an antibody test. Just because you want to have a history of what's happened in your own company, but also the CDC does recognize that antibodies are likely to provide at least some short-term protection. The CDC, in all fairness, also warns that there is no reliable evidence at this point, but it's likely. It's like the flu. If you got it once, you probably don't catch it right away again. So it's fair to assume that people with antibodies are less likely to be reinfected and or to transmit the virus, and maybe these should be the people who should interface with customers if you absolutely need to do that. But the bottom line again is that there's a general problem regardless of the platform. There's no easy solutions for routine at home or at work testing yet. All right, so now let's have a look at the aspects of the various tests. The molecular tests are often run in clinical labs, which are very well equipped, like this environment here, and they are run on clinical analyzers, such as this one. This is a popular one by Roche. These are big machines, they're very expensive. They have a high throughput, so you can do hundreds of samples in parallel. They have the best accuracy of what you can find on the market, pretty much. Roughly 98% sensitivity and 100% specificity. The test takes 3 hours from beginning to end, but of course that does not include the logistics of getting a sample, bringing it to the lab, and then sending the results back to the physician and to the patient. An alternative to these clinical analyzers for molecular tests are these benchtop type of instruments. So on the left is one by Cepheid. One puts a swab into this little cartridge, you close it, and within, I'm not sure exactly, maybe 30 minutes, something like this, 45 minutes, you get the result. And same for this Abbott device, which has actually 2 cartridges, a blue one, an orange one, and yet a third one to transfer materials from the blue to the orange cartridges. So these are actually quite attractive options. Again, they're not allowed right now for use at home or even by an employer like us, for instance, but hopefully they will become available soon. This is probably a good option altogether, but there is still a catch. And these molecular tests are only as good as the sampling method. So that's very important to understand, right? So the molecular test might be good, but the sampling method is not. So the CDC recommends these famous nasopharyngeal swabs, which are still considered to be the gold standard. But what's a nasopharyngeal swab? Well, you need to insert a very flexible swab into the nostril. And let's look at this diagram here. So the swab goes into the nostril. It has to go horizontally all along the top of the palate without touching the delicate structures of the turbinates, all the way to the very back here, and this is the nasopharynx in blue. So it's pretty much at the height of your spine and of your ears. So needless to say that it's not a convenient procedure. It's not nice, and it's prone to errors when you actually collect the sample. Saliva might be a good substitute. There start being a couple of tests that use saliva. It's not fully demonstrated yet, but hopefully data will come up showing that saliva actually works as well as nasopharyngeal swabs. But for the moment, this is the gold standard. Then for the antigen tests, you require pretty much the same type of hardware. You still need a swab. You need a desktop instrument, which are becoming increasingly small, and you need a cassette. Like here, on which you put your sample and then you insert it into the reader. These tests, well, as far as I know, there's only 2 of them that have been authorized by the FDA, one by Quidel and one by Beckman Dickinson. The sensitivity is of the order of 85%. The specificity is about like what's happening at Quidel. Their stock has tripled in the last 3 months. And then there's the antibody test. So the antibody test can be run on anything from the sophisticated clinical analyzers that I had earlier on to these benchtop type of instruments to just a naked eye readout like here on this picture. Here you require blood that's collected typically 3 weeks after the onset of symptoms. If you run this on a clinical instrument, then the test is really sensitive and specific. And if you want something that you can be read with the naked eye, it's portable, it's very convenient, it's very cost effective, but the market, as you know, has been flooded with tests that have very poor performance. So the FDA, unfortunately, that's, by the way, an unfortunate outcome of the emergency use authorization. So the FDA is compensating by requiring a medium to high complexity lab to review the test and perform the test. And that really defeats the portability of these devices. And the FDA is currently withdrawing more than half of the tests that they previously authorized just because the quality is not there. So this table tries to capture everything I said in terms of sensitivity and specificity. Molecular tests are highly sensitive, highly specific. There's also, by the way, a very good concordance between different manufacturers. So you should not worry too much as, did I get a test from company A versus company B? But what they do is that they require, again, a well-performed swab. So you need to have trained personnel that can do that well. The antigen tests are 85% sensitive, 100% specific. The antibody tests, that's interesting. So the manufacturers claim 98% sensitivity, and these are big names, right? Like Roche, Abbott, and so on. But there's some literature that says that it's probably closer to 85%. So where the truth is, I don't know. I just wanted to give some color to these numbers. And then there's the portable rapid tests. The FDA requires greater than 90% sensitivity and greater than 95% specificity. But as I said, they're pulling out right now 45 out of 77 previously authorized tests because the performance is just not there. So in summary, and I like this quote from the Science magazine, what everybody wants is a test to be cheap, accurate, and fast, says this gentleman, Jeffrey Bird, a lab specialist at the University of Washington, Seattle, but you can only ever have 2 of these. And that's very true. But I would add that the challenge is not only to make a test that's cheap, accurate, and fast, but also one that requires a simple sampling method, so not the nasopharyngeal swab, and that can be amenable to routine use at home and on the workplace. So that's the holy grail. So now let me switch tack for the next few minutes. By the way, I will not need the full half hour. So where do we come in? Well, here is another use case that we have not mentioned, and these are low-income populations. This is a picture I took in Nigeria 2 years ago, and it's market day, and it's complete chaos. So how do you manage COVID-19 in this type of environment while us in the United States are barely having our act together despite our wealth, our scientific expertise, our industry, our communications, and so forth? And I don't know, it's going to be a dramatic problem. I'm very concerned about these countries. And one does not need to go necessarily all the way to sub-Saharan Africa to have problems. Just, a few minutes away from San Diego in Mexico, I think that the situation is going to be very difficult to manage as well. Traditionally, we've been working on these neglected tropical diseases. One example is Loa, where you can see the parasitic worm migrate under the skin, sometimes come out of the eye. Few people have heard of it, but there's 10 million cases in sub-Saharan Africa. Another disease we're working on is river blindness. It's also due to a worm. They lodge in the retina and cause permanent blindness after several years. And the third one is lymphatic filariasis, due to yet a third type of worm that goes into the lymphatic system, blocks the lymphatic system, and leads to these massively enlarged limbs. So in the process of working on these tests, on these diseases, we've developed an ultrasensitive platform to detect antibodies. And our motto is really high quality and low cost. Our antibody tests are up to 30 times more sensitive than similar ones made by others. One thing of which I'm quite proud of is that for one of these diseases, our test is 15 to 30 times more sensitive than a test made by Abbott, and that is a huge feather in our cap. So since we have this highly sensitive and specific platform, it was natural for us to retrofit it to detect COVID-19. And we've developed a prototype, which we sent 2 months ago to the New York Blood Center for an independent evaluation. And it came back being 93% sensitive, and this is great. The FDA requires at least 90%, and 100% specific, and again, the FDA requires at least 95%. So these are some mock samples made with blood. These are not the real samples, but they give you a sense for what the test looks like. Well, in the meantime, we have improved the quality of the tests, and right now we're on the cusp of freezing the assay design. And we're also in the process of planning a clinical trial to submit data to the FDA in view of this emergency use authorization. Our objective primarily is to make tests available to low and middle income countries, LMICs for short. And the tests will be initially made available for free so that the countries can validate them and understand how to best use them. I should add that we have a large network, both with the local ministries of health, with WHO and other organizations, and I think that will help us disseminate the tests in those countries. An ideal scenario would be one in which for each test that is sold in the United States, another one will be donated in a low to middle income country. So basically the net profits from domestic sales would fuel donations in other countries. It is an antibody test, and as I said, these I think have their best place in supporting public health decisions. I should also add that many countries do not have sufficient infrastructure for molecular tests. I've seen dispensaries, for instance, in which there is nothing. There's no water, no electricity, no drugs, nothing. There's no band-aids, there's no alcohol, there's just a bed, that's it. And so in those circumstances, right, it's unrealistic to expect that these countries will be able to perform molecular tests at the scale they need. So antibody tests may find some uses for individual case management, even if they're suboptimal for that. But we realize also the limitation of the antibody test. So our future plans are to develop a salivary test, maybe something like this, that you would insert into the mouth, that could be used at home and in the field. We want to be able to detect both antibodies and antigen on the same test strip. Now, this is extremely challenging. It's not an easy nut to crack, but it's also extremely needed. So we'll see what we can do in this respect. This is the last slide. One of the reasons, or maybe the main reason for which we're working on this type of diagnostics is that I really genuinely believe that you can have, that a little money goes a long way. You get a lot of bang for your buck. So just to give you a sense, right? The anti-blood test was developed completely through a generous network of philanthropists in San Diego. We did not even need that much money, something like $200,000. Now we want to jumpstart the use of these tests by sending them to these countries. The manufacturing costs are of the order of $2 per test. So with $100,000, we should be able to start distributing the first 50,000 tests. And we estimate that after that, things should become self-sustainable. The salivary test is a different thing. Again, it's a very ambitious program. Will require much more than $100,000, but we first need to generate proof of concept data just to show that we're on track, and we'll get there when the time is right. And this is pretty much it. I wanted to thank all of you for just attending this seminar. Again, I wish I could see you face to face and not talk just to, a blank computer. Please do visit us at DDTD.org. Please always feel free to reach out to us. We're always available. This is my email, marco.biamonte@DDTD.org. You can also reach out to Melissa, our Executive Director of Development, whom you've just heard, melissa.chalmers@DDTD.org. We're only an email away, a phone call away. We're happy to show you our lab. Just keep in touch with us. And with this, I will hand it over back to Kaitlyn and Mike, I assume.
Thanks, Marco. We're going to transition to some Q&A. And the first question is, how do we know the most reliable place to get testing done? And sort of who can you trust to go get a test done? And that may depend on whether you feel like you have symptoms, or maybe you were sick previously, and you kind of want to figure out whether you have antibodies. What's the best place to go get a test?
So yeah, so it's a very good question, right? And it's a question we all want to know. So the answer is simple. Just go to our major healthcare providers. All the molecular tests have comparable performance. Some of them might be just a little bit better than others, but it really does not have a lot of clinical significance. So any molecular test will be good for you. The antibody test, make sure that you, again, go to a major provider that will send your sample to a clinical lab. For what I've seen, I would, the only small test, portable test I trust right now is ours, but it's not marketed yet. So don't do those yet.
So there was a question about if coughing transmits the virus, why wouldn't a cough type sample be a way to test, or is that a potentially a viable way to collect a test?
Yeah, that's a great question. So again, so this is a so the short answer is yes, right? So these molecular tests were initially developed with these nasopharyngeal swabs. People are trying to develop them with saliva. They're known to work with sputum. They're known to work with also more complex matrices like a bronchoalveolar lavage. So yes, they're getting there. The problem is that it's difficult to, I guess, get enough data quickly enough to demonstrate 100% that the sensitivity is the same as for these nasopharyngeal swabs that have been widely used.
And then there was a question about how to find out what type of equipment is used for the test that you're receiving. Do you find that that's easy enough to find out? Would you just ask the provider what type of machine are you running the test on? Is that easy information to find out?
Yes, well, I don't know if I don't know I don't know that answer in detail, but that's exactly what I would do. I would ask my medical doctor. But again, if things are being run in a clinical lab in the US, probably all the tests are equivalent, and it's unlikely that there's going to be major differences from test to test. And again, I've been maybe a little heavy-handed on this message, but I think for molecular tests, it's the sampling method that makes the difference, not so much the test itself.
There was a question about false negatives, and a question about maybe a 30% false negative rate. And so the question is, if you get a negative test, but you're still not feeling great, how long maybe should you wait to go get another test? Should you wait a week or so, or to see if maybe antibodies are present, or how long should you wait between tests?
Yeah, that's a great question too. I'll answer slightly besides the point, but that's a message that I try to keep giving to WHO over and over. Of course, there's always the pressure to run only one test, and to think that if you run one test, you're going to that every test is a miracle test, right? That they all work perfectly well. I think that running two tests in parallel is really your best option, and then you're not in doubt. So if you can run a molecular test as well as an antibody test, then I think if the two answers are concordant with each other, you will have your answer with certainty. Does that answer the question, Mike, you think?
Yeah, I think so. So, and this is I'm obviously not an expert, but I would say if you're uncertain, go get another test. And the difficulty is sometimes the time and the expense of doing it. But if you really want to know, or if you think you have symptoms, then go get another test.
That's right. Also, the popular wisdom is that the median time for between the time of infection and the time at which the symptoms appears is 5 days. So 50% of the people will develop symptoms within the first 5 days, but another 50% of the people will develop symptoms between day 6 and day 12. And by day 12, pretty much everybody has developed the symptoms, if they will. So maybe that's also a different metric that could be useful to the person who's asked the question. If you're not positive by a molecular test after day 5, maybe wait until day 12.
So I'll give you this question. You may not want to answer it, but there was a question about just protecting yourself from others. If you wear a face shield, you also need to wear a face covering. So that may not be your area of expertise, but I'll ask anyway, and you can feel free to defer to others if you'd like.
Yeah, thanks, Mike. So yeah, that's right. So that's not my area of expertise, and it, of course, I have the same questions for myself, and you cannot quantify of transmission. But there's a couple of thoughts. We still know that the primary route of transmission is through, talking and emitting droplets of saliva. It's not the only one, but if you wear a mask, you're protecting others. You're not necessarily protecting yourself, but you're protecting others, and I think that's important. And also, I think it's important to stress that even, and I don't know what the numbers are really, but let's assume for a second that the mask is not a perfect protection. Decreases the rate of transmission by twofold only, right? Then even that is very important from a public health perspective, because decreasing the amount of transmission by twofold might be the difference between having a hospital overloaded or not, or between having an economy that can work or an economy that doesn't work. So I very strongly encourage people to wear a mask. It doesn't take a lot of effort, and I think it's the responsible things to do.
So there was a question about the timing of when you think the low-cost, high-specificity antibody test that you guys are working on might be available in the U.S. So realistically, when do you think something like that could be available, where you could have a home use application or something low-cost, easy to administer that would give an accurate result?
So we're racing as fast as we can, of course. We still need to perform the clinical trials. That probably takes a month, and some I don't know exactly because a lot of things are not under our direct control. Then we submit data to the FDA. And I should also explain that when you submit things to the FDA, you have two different paths. You can submit for this emergency use authorization, which is a sort of low bar, or you can submit to ask for a device to be approved, at least at the point of care. And you have to demonstrate that people with no training, essentially, or minimal training can actually run the test efficiently. That's a different order of magnitude. So I think we'll submit to the FDA for the emergency use authorization within the next 2 months. And then we can start selling it. But again, only in labs. And I'm not sure how much time the FDA will require to review the more complex application. I know that they're swamped though.
Well, I think we've addressed the majority of the questions. If folks have follow-up questions for Marco or Melissa, they're more than willing to engage with you and happy to answer questions beyond. I want to thank both Marco and Melissa for the investment of time and all the coordination that goes into putting these things together. Thanks also to Caitlin from our team and Jesus from our team who worked so hard on the technical aspects of putting this together. But these are the amazing things that are going on in San Diego all the time, and DDTD was doing amazing things before COVID-19, and this pivot is really exciting. So thanks again to Melissa and Marco for your time and investment, and for sharing with us. And hopefully this helps us be better informed about the tests that we might be receiving and where to go get them and what they mean. and hopefully that helps us in making better decisions. So thanks a lot, Marco. Thanks, Melissa. And with that, we'll wrap things up.
Thank you, everyone. Again, feel free to reach out to us for any reason or any feedback. Mike, thanks a lot. Kathleen as well. Goodbye, everyone.
Bye-bye.